Good afternoon and welcome back, everyone. I hope you had your coffee and your tea, and now you're feeling even more energized because the last session of the first day is here. But before we start, I would like to see the answers for our third question that was in the Slido platform, because we truly had very good the last session. And the third question was, which career model provides higher job satisfaction for a scientist? And as we can see, well, it's almost 50-50, I would say. Academic or industrial, academic wins by slight percent, but I would say it's almost a tie. So I guess it truly depends on the person. So thank you very much, each and every one of you who went on this Slido platform and answered all three questions. And we got the feel of the answers of these questions. So thank you. Now, about this last session, which are pitches of Alliance4Life's members to industry and ESRs. The Alliance is interested in developing an industry relationship platform as an informal communication and meeting place. The Alliance4Life's members take the advantage of investments made in the past decade into the state-of-the-art infrastructure in Central and Eastern Europe. Therefore, the short pitches are focused on open access to the infrastructure and related services, but also on scientific collaborations and technology transfer. Each speaker in this session will have 5 minutes for the pitch. And to keep the session quite, I would say, dynamic, I'm afraid that there will be no time for questions. However, if during presentations you have any questions, feel free to write them in chat here in this platform by addressing the person you would like to ask the question to. And for all the speakers, I would kindly ask to have a look in the chat during this session just to check if there are any questions addressed to you. And if there are, feel free to answer them in the chat or get in touch directly with the person who is asking. So I think let's start the last session of the day. And first speaker of this session is Zlatoše Novotna, who is Alliance4Life coordinator. Zlatoše, good afternoon and welcome.
Hello, good afternoon to everybody. Allow me to introduce Alliance4Life shortly, as Martin already announced. It's my pleasure to welcome you today on behalf of the Alliance4Life, and we will very gladly show our offer to partner companies as well as to our young researchers. I am also head of strategic partnerships working for CETEC, Masaryk University in the Czech Republic, and I'm very proud to be coordinator of the Alliance4Life. So next slide, please. Who we are. We are 12 progressive life science institutions and universities from Central and Eastern Europe. And we all know that our region belongs to the less performing regions of the EU in terms of research and innovation. Therefore, we joined forces to trigger a change. And our activities have been supported with already 2 projects from Horizon 2020. Next slide will show our members. We selected our members based on a comparison of scientific performance. So we are proud to cover the best performing life science partners in our countries and mostly also the largest universities. The Alliance is led by CETEC, located in Brno. And as you can see on the map, from north to south, our members are located in Tartu, Riga, Vilnius, Lodz, Brno, Bratislava, Budapest, Ljubljana, Sofia, and Bucharest. So next slide, please. I will tell you what is our vision. We are open to introduce necessary changes. We inspire each other to improve together, and we are motivated to create the best possible conditions for excellent research with impact on innovation, of course. Some of our members have a well-established tech transfer office. Some of them are in the process. but you can be sure there is a lot of talent and motivation in our region. And as was mentioned already, we have a state-of-the-art equipment, which is for sure our advantage. Next slide, the most important, what we offer. We offer a win-win approach to the industry. We offer access to so-called pockets of excellence, as we call it, in biomedical sciences. We offer to explore opportunities of joint research projects with our best researchers, and we will organize thematic mini conferences with them and with you if you are interested. We offer access to our equipment and services, and for this we have created an electronic database of core facilities and related services, which will be soon on our website. And we also prepare open access calls. And of course, being part of our relationship platform means that you have a priority access to our retreats and events, similarly as today. So we are happy to stay in touch and shape our activities to meet your needs and expectations. What we offer to PhD students and young researchers: of course, we offer study stays, internships, and postdoc positions and our institutions. So you are also very welcome to have a look on the following pitches. And important information is that we will organize further events, trainings, and retreats, so you can take part. The next event will be Skills Academy in April, focused on soft skills, and we hope it will not be the online event as this one. So next slide, please, shows our Twitter account. Yeah, please, the next slide. Yes, you can follow us on Twitter or on our website to be updated about our news. We will be happy that having you as followers. And next slide just shows my contact information. That's all from me for now, and let me wish you a good pitching session.
Thank you, Zlatoše. Almost perfect in time. Amazing. Thank you. Thank you for giving this inside view in Alliance4Life. Now let's go to the next speaker, who is Daniela Trshova. She is Business Development Manager at Central European Institute of Technology, Maastricht University. Daniela, welcome. Please, the floor is yours.
Good afternoon, everybody. My name is Daniela Trshova. I'm a business development manager, and I would like to introduce you to research at the Institute CETEC, Maastricht University. Next slide, please. The name CETEC is an abbreviation of the name of our consortium, which is the Central European Institute of Technology. This consortium was established in 2011 on the strong foundation of 6 prominent universities and research institutes in Brno, Czech Republic. These partners are from the area of plant research, human and veterinary medicine, biology, and material analysis. One of our strongest selling points is the interdisciplinarity from combining the knowledge and resources from life and material sciences. I will now talk about the biggest partner in terms of labs and people, the Maastricht University, shortly CETEC MU. Next slide, please. Our experiments are focused into 4 research programs in following areas. In health, there is the program Molecular Medicine, dealing with nucleic acids and protein research, cancer biology, and infectious diseases, including the COVID-19. And the program Brain and Mind Research, covering brain disorders like epilepsy. In plants, there is the program Genomics and Proteomics of Plant Systems, experimenting in developmental and stress biology of plants. And the last one is the program Structural Biology, studying living organisms at the cellular, molecular, and atomic levels using advanced microscopy techniques. Next slide, please. A strong reason for cooperation is our 11 core facilities spread on 25,000 square meters, equipped with high-end instrumentation, one of the best in Central Europe. Core Facility Genomics provides services in next-generation sequencing, microarray analysis, and flow cytometry. The data from genomics lab are processed in the bioinformatics facility. The proteomics lab covers all the steps of the mass spectrometry-based proteomics analysis. Plant sciences provides access to 15 walk-in-growth chambers called phytotrons and 10 greenhouses, delivering a defined environment for controlled plant growing. Cellular imaging provides services in light microscopy for complete studies in modern cellular and molecular biology. Cryo-electron tomography addresses structures like cells and organelles, while cryo-electron microscopy is applied to study single particles, mainly large macromolecular complexes. The NMR facility can measure proteins, nucleic acid, and carbohydrates, but they can also manage the analysis of materials. The BIC facility provides services for the biophysical and structural characterization of biomolecules. The X-ray lab can show the shape, size, and the 3D structure of molecules, for example, in pharmacology. The facility Nanobiotechnology uses atomic force microscopes to study tissues, biomolecules, or cells. And the last but not least, the MEPHIL lab provides human magnetic resonance imaging and EEG measurements for neuroscientific research. Next slide, please. One way of cooperation can be the joint research with one of our research groups, or you can approach us through our core facilities, which are available through the online booking system on the open access principle and are available both for academic and commercial users. You can book just the instrument time, order specific training for your employees, or you can order a complete service beginning with the sample preparation and ending with the data analysis. Another way is to exchange students and employees in some mobility program. In case you want to license an existing intellectual property or want to invest in a spin-off company, the terms of the agreement will be reviewed by our Masaryk University Technology Transfer Office. Next slide, please. On this slide, there are some examples of a successful cooperation with industry. The market area of common interest with the company is usually the medical diagnostics, pharma industry, biotechnology, chemicals, nanomaterials, or agriculture production. Next. If you're interested in any of the research areas at our institute mentioned before, you can reach me at one of the listed contacts. I will connect you to our research groups, core facilities, or the TTO. I'm looking forward to our future cooperation. Thank you.
Once again, perfect timing. Daniela, thank you very much for your presentation. I would like to remind all of you who are watching right now that if you have any questions during these presentations to any of our speakers, please feel free to write these questions in chat. And after presentations, the speakers will have a look in the chat and answer them directly or in the chat as well. So our next speaker is Oswald Spugovic, who is Director at Latvian Institute of Organic Synthesis. Oswald, good afternoon and welcome, please.
Good afternoon to everyone. I hope you can hear me. Thank you, Martin, for a kind introduction. Yeah, I'm Oswald Spugovic, Director of the Latvian Institute of Organic Synthesis, and I'm going to introduce you to the leading drug discovery center of the Baltic region. Next slide, please. We are here since 1957, and currently there are a little bit more than 240 employees at the Institute. We have wide collaboration network with industry and universities, and we are proud to be a birthplace of 18 original medicines and more than 100 technologies of generic drugs. And last but not least, we are the top-ranked research organization in Latvia, according to the assessment by International Panel of Experts. The assessment was done in 2020. In 2020, we had a total funding of almost 14 million euros, and we are proud to say that 1/3 of this funding comes from private sources. That means collaboration with industry. Next slide, please. And this collaboration with industry shows that we are operating worldwide. So almost half of the funding comes from the European Union, a quarter comes from US, And then there is a collaboration with UK-based companies, Swiss-based companies, and also with our local pharmaceutical companies. We do services for biotech companies as well as for medium and large pharmaceutical manufacturers. And this includes discovery of original drugs, manufacturing technologies, technologies of generics, as well as analytical chemistry services. But the most important thing in this slide is that 75% of our customers work with our institute for more than 5 years. Next slide, please. What we do. Medicinal chemistry services include synthetic hit-to-lead, lead optimization studies, in silico drug design is widely applied, structural biology, fragment-based lead discovery, as well as we are introducing natural products research to have new core structures. Pharmacology and bioanalytics is, of course, efficacy testing, ADME testing, but also this basic research in pharmacology allows us to identify new drug targets. It allows us to discover mechanisms of action and repurpose existing drugs. Manufacturing technologies include route scouting, upscale, kilogram scale synthesis, impurity profiling, and GMP compliant analytical chemistry services. So far, the most successful areas have been anti-infectives, cardiovascular, CNS diseases, and anti-cancer medicines. Next slide, please. The last marketing authorization of a drug discovered at the Institute was in 2014, and now the holder is our partner company, Acrotech. In 2019, we have our partner, local pharmaceutical company, Grindex, has finished phase 1 clinical studies of a cardioprotective drug candidate, methyl GBB. Next year in 2020, a US-based biotech company entered in phase 1 clinical trials with a candidate drug for treatment of neurodegenerative diseases. But that's not the end. We have more than 10 collaborative and proprietary projects in our early stage development in a phase from lead compounds to CD. Next slide, please. One of the features, how do we work, is fastness, efficiency, and we are also a reliable partner as it was shown by the lengths of collaborations. Just an example, in 2016, we started collaboration with a US-based biotech company, and we were providing chemistry and pharmacology services for this company. In 2019, we ended up in a patented lead series. The hit-to-lead phase was over. And next year, in 2020, the same company entered clinical trials with a drug candidate developed at the Latvian Institute of Organic Synthesis. Currently, we have about 38 collaboration partners in last year. And next slide. And of course, anyone can be a new collaboration partner. Just contact us at our website, and you will be directed to the research groups, to the experts of area of interest. Thank you.
Great. Thank you, Oswald, very much for your presentation. Have a good evening, and thanks for joining us today. Let's move forward, and our next speaker is Martin Schauer. He is Business Development Manager at International Clinical Research Center. Martin, I hope you can see me and hear me, and I can see you. Hello and welcome.
Hello. Good afternoon, everyone. I am a representative of ICRC, and I am a business development manager working at the department with interesting name, interesting acronym, which is BAR, which means Business and Academic Relations. Okay, let's move on. Next slide. And why I am showing the hospital? Because ICRC, we are a part of the hospital with name St. Anne's University Hospital in Brno, Czech Republic. The hospital has a long tradition, and it is interesting that Johann Gregor Mendel gave his foundation of genetics in the same place as the hospital is placed. Please, next slide. Here is the timeline of the hospital, and the last point here is the establishment of our institution. It was in 2011. Please, next slide. ICRC, well, what does it mean? It's the acronym of International Clinical Research Center, and you can see here our motto, which is creating the future of medicine. Okay, next slide, please. As you already seen, the ICRC was founded in 2011, so it celebrated its 10th anniversary last year. Since then, there we have more than 120 grant projects and around 500 clinical trials. From the very beginning, our strategic partner has been Mayo Clinic, which is a very important company worldwide. The research is focused on basic, preclinical, and clinical research. We have something around 500 employees, and we have over 1,500 scientific publications. Okay, next slide. Our results, as you can see, we have 2 nation patent granted and 2 American patent granted. Right now we have 12 patent application and 12 utility models, but very often it is a result of projects. Okay, next. As I mentioned before, our research is focused to clinical research, basic and preclinical research. At this slide, you can see our research groups. In clinical research, I can mention, for example, stroke, oncology, cardiovascular magnetic resonance, and so on. In basic and preclinical research, I can mention cell and tissue engineering facility, protein engineering, cancer plasticity, and so on. You can see that some groups are marked, and it means that these groups provide also core facility services. Okay, next slide. At this slide, you can see how we cooperate with industry, and there are also a few important companies we cooperate with. Okay, how we collaborate? We collaborate through joint grants, preclinical studies, clinical trials, contract research, and launch of devices. And from companies we collaborate with, I can mention, for example, GE Healthcare, IQVIA, Philips, AstraZeneca, and Pfizer. Okay, next slide. Thank you all for your patience, and I am looking for our future collaboration. Thank you very much.
Martin, thank you very much for your presentation. These 5 minutes are flying by really fast. Thank you, have a good evening, and thanks for joining us. Our next speaker is Silvia Pastoreco. She is professor and director at Biomedical Research Center of the Slovak Academy of Science. Silvia, good afternoon and welcome.
Good afternoon. Thank you very much. I'm very pleased by the opportunity to introduce you the Biomedical Research Center of the Slovak Academy of Sciences and provide you a brief insight into some of our unique facilities. Next slide, please. Yeah. BMC was founded in 2016 by merger of 4 institutes and now consists of 5 institutes performing basic and translational research in a broad range of topics, mainly in virology, oncology, endocrinology, genetics, and neurobiology. Next slide, please. Our human capacities involve about 250 researchers and 50 PhD students. And the research infrastructure includes 13 specialized laboratories and facilities, such as research clinic for outpatient healthcare, center of physical activity with tailored intervention program for seniors and diverse groups of patients, biosafety level 3 laboratories for work with dangerous pathogens, and biotechnological and analytical laboratories. in this on the last two facilities. If you can show me the next slide, please. The basic as well as contracted research of highly pathogenic viruses and microorganisms is performed in the BSL-3 laboratories, consisting of laboratory for cultivation and molecular studies of dangerous pathogens, the animal facility for in vivo experiments, and laboratory for GLP production of Q fever vaccination antigen. Next slide, please. In this core BSL-3 laboratory, we perform, for example, isolation of viruses and their biobanking within the globally largest virus archive, EVA Global, in which we take part as a founding partner, contributing by biobanking of hantaviruses, tick-borne viruses, coronaviruses, and other dangerous viruses. These products or these viruses and derived products are available to both academic and industrial partners. We also validate different tests, including qPCRs for SARS-CoV-2 in collaboration with the Slovak company Multiplex DX. We also do virus neutralization tests and testing of antiviral properties of different materials. Next slide, please. The other very unique facility is located in Eastern Slovakia and is dedicated to R&D of biopharmaceutical. It has high-tech premises for work under aseptic conditions according to GMP requirements, and also validated assessment of composition, safety, and purity of bioactive compounds in GMP certified regimen in compliance with the European Pharmacopoeia. Next slide, please. This consists of three sections. Section A is dedicated to research in eukaryotic system, prokaryotic system, research production of vaccines, growth factors, antibodies, media preparation, etc. Then next slide, please. Section B is dedicated to development in active pad work with living viruses, cultivation, propagation, and in part B, it's an inactive part with inactivated viruses and specialized purification processes. And also there are some additional spaces. Next slide, please. And the most frequently used is the section C for analytical processes, where we do have specialized laboratories for analytics using broad spectrum of quality control methods, chemical, physical, microbiological, serological, virological, and molecular biological. And there we also have a lot of partners, for example, hospitals, quality control of infusions, then pharma companies for the control of raw materials and products, blood derivatives, drugs, etc., and also materials for their bactericidal or antiviral properties. And we also cooperate with other partners, for example, for biomonitoring of spaces. Next slide, please. All these facilities are open for collaboration with industry and also academia, and selected spaces in this R&D facility are opened also for rent for smaller companies. Additional information can be available on the website of BMCSAS, and I thank you very much for your attention.
And thank you, Sylvia, very much for your presentation this evening at Alliance4Life's event. Thank you. Next speaker is Maria Cicalevska-Studzinska, who is postdoctoral researcher at Medical University of Łódź. Maria, good evening and welcome, and please, the floor is yours.
Hello and welcome. Thank you for the possibility to present our lab, which is a central scientific laboratory core lab. We are a core facility that provides services and expertise while maintaining the highest standards by highly qualified staff. For more than 10 years, we've been implementing scientific and research projects in the fields of molecular biology, genomics, and proteomics. Next slide, please. Our core lab services include providing access to high quality scientific infrastructure, consultation and assistance in planning and designing of experiments, analysis, visualization, interpretation of the obtained results, and scientific assistance and support in preparing grant application process. Our strong points are individual approach to each project, our knowledge, scientific potential, and the many years of experience for carrying out research in interdisciplinary projects. Can I have a next slide, please? The scientific offer of Core Lab is divided into three following research areas, which is gene, protein, and cell. And the gene section is divided into DNA and RNA analysis. And in DNA, I'll just mention a few examples. We do the study of mutations and polymorphism in genetic diseases, and the study of animal DNA presence in food, cosmetics, packaging, or as far as halal is concerned, for example. In RNA analysis, we do the studies on disease-related changes in gene expression, monitoring of course of treatment, and influence of tested substances on the expression of selected genes. In the field of protein analysis, we can check drugs and hormone levels, chemicals and toxins, allergens and mycotoxins. And as well as in analyzing the food, we can check the parameters indicating classification of food products. And looking from the wider perspective, in cell, we conduct research on chemicals and processes as far as cytotoxicity studies. Analysis of microorganisms involved in technological processes. This is biomarkers identification and the structural and functional diversity of microorganisms in studies. Can I have a next slide, please? I'll just say a few words about the equipment we use in our research. So basically, we have the droplet digital PCR system, which is mainly used for rare mutation detections and absolute quantification of nucleic acid, and 7900 real-time PCR, mainly for gene expression levels, microRNA expression and telomere length. In the field of cytometry, we use cytoplex, which is apart from regular cytometric function, it is able to detect the microparticles. Next slide, please. The next one is the next platform is the MagPix, based on BIT technology, and the Multina, which is an alternative for typical electrophoresis system for DNA and RNA analysis, and the Victor, which is a automatic multitask microplate reader that allows us to use many absorption, fluorescent, and luminescent markers. Next slide, please. Medical University of Lodz has few more units that provide business services for example, animal research facility that is entitled to conduct biological research in vitro and in vivo using animal models and cell lines. And they offer training courses in the field of experimental procedures on laboratory animals, providing test equipment to companies and for projects, and help in designing of experiments on animals. And there is also Dynamo Lab, where they can carry out complex analysis of human movement and physical performance, as well as training of guide, posture, and, for example, checking the lung capacity. I highly recommend checking the websites. And next slide, please. Please do not hesitate to contact us for more detailed information, and we are happy to cooperate. Thank you.
Maria, thank you. Thank you very much for your presentation as well. Have a good evening. We are precisely in the middle of this session, right? We have 6 speakers already presented their presentations, and now we have 6 to go. What I would like to say is that if you have any questions at all, please write them in the chat, and speakers will try to answer them directly. So let's move forward. Our next speaker is Siniša Skokic, who is Laboratory for Regenerative Neuroscience. He works at University of Zagreb School of Medicine. Siniša, good evening and welcome. Sorry, you have to unmute, please.
To unmute. Okay,
Thank
thank
you.
you. Thank you for the introduction and welcome to this presentation. My name is Siniša Špokić and I will present industrial collaboration opportunities at the University of Zagreb School of Medicine in Croatia. Next, please. The University of Zagreb School of Medicine has recently celebrated its 100th anniversary with a millennial photo shown here on the left, which makes us one of the institutions with the longest tradition in medical training and research in Southeast Europe. We are also one of the largest research institutions with over 800 employees, more than 500 of which are involved in research. Many of our employees share their workload between academia and one of the 5 clinical hospitals in Zagreb. As a result, our institution alone accounts for almost a quarter of total Croatian research production. Next, please. With departments spanning from biology and chemistry all the way to neuroimaging, and many of our teaching staff doing research in one of the university clinics, we can support biomedical research projects at all stages, from basic to clinical studies. The examples below highlight some of the more recent examples, whereby School of Medicine was either a partner or the leader of the international consortium. As you can see, our involvement ranged from processing in vitro assays to designing and performing a large animal study in collaboration with partner institutions, and up to a phase 2 clinical trial. We are thus the perfect partner for anyone looking to translate their idea from the laboratory to the clinic. Next, please. So what can the University of Zagreb School of Medicine offer you to boost your idea and help you hit your target? Let us draw your attention to the 5 laboratories that offer commercial services. Starting from bottom left, the Laboratory for Proteomics hosts equipment for mass spectrometry and liquid chromatography and can detect biomarkers of early pathology or validate the purity of your compounds. At the Laboratory for Intercellular Communication, we can perform microbiota analysis and many other tests related to compound profiling, such as their antibacterial or antioxidant activity, cytotoxicity, etc. Laboratory for Functional Genomics specializes in next-generation sequencing, whether it be on the whole exome or a clinical exome, and can determine diagnostic gene panels specific to the disease of your interest with the highest expertise in the field of neuroscience. These 3 laboratories operate within our Center for Translational and Clinical Research, founded in 2009 as a joint organizational unit of the School of Medicine and Clinical Hospital Center Zagreb. It aims to facilitate and streamline collaboration between the School of Medicine, clinical hospitals, and industry. Above the illustration, we have a laboratory for electron microscopy and laboratory for regenerative neuroscience. The former looks deep into the cells with immunogold microscopy, which can be employed for assessing nanoparticle and drug delivery, localizing protein synthesis, or detecting DNA or RNA viruses. The latter is dedicated to preclinical research, specifically longitudinal in vivo imaging of small laboratory animals, such as mice or rats. It is a unique facility in the region and also unique within the Alliance4Life consortium, allowing to not only image the animals, but also house them, perform any surgical procedures on them, or prepare any biological agent for their treatment, and all of that within the same facility. This allows us to offer a turnkey solution to our clients, starting from protocol development through any and all manipulations, either on cells or on animals, and all the way to the data analysis. Next, please. Like many other partners in the Alliance4Life consortium, we are open to any form of collaboration, from providing educational services through collaborative projects to performing contract research. You will also notice that some of the services on offer are not unique to us. So why choose University of Zagreb School of Medicine? We believe that our key advantages are our direct link to the clinic, materialized through our Center for Translational and Clinical Research, and our size and experience. Many more labs are able to join in as required in order to grease the research gears and accelerate your project. Finally, let me point out that it is ultimately not the equipment, but the people that count the most. In all of the presented facilities, we have experts who have been perfecting their skills for at least 10 years. In some cases, like electron microscopy, we have over 30 years of accumulated know-how. But the know-how is not exclusive to the scientists. Our technology transfer experts have actually transferred from the pharma industry to the university and have valuable insight into the industrial aspects of their collaboration. Next, please. So for any inquiries, if you're interested, please contact us at kto@mef.hr. We will be glad to meet you and discuss your project ideas, and thank you for your attention.
Senisha, thank you very much for your presentation as well, and thank you for joining us. As you can see in chat, I would like to also remind you that during speakers' presentations, please use view options side by side. Then you will see the whole presentation and nothing will be left out. Thank you. And next speaker is Simon Botond. He is at Department of Conservative Dentistry, Faculty of Dentistry, and Karoli Rach Doctoral School of Clinical Medicine, Semmelweis University. Simon is here and he is, I think, joined us.
Yes,
hello, Simon. Welcome.
Good afternoon, everyone. My name is Botond, and I'm a dentist by profession, and I represent Semmelweis University with today's presentation. Semmelweis is Hungary's leading medical university, which is very committed to supporting translational innovation. My topic is related to human identification. Related to human identification. We managed to prove that the palate is suitable for human identification, for which we started to develop a three-dimensional pattern recognition software called Palatoprint. On the next slide, just to give a brief introduction to you, we believe in changing the way of human identification. Currently, 3 primary identifiers are used by the authorities for identification: the fingerprints, DNA, and dental records. Of course, these primary identifiers used so far have their pros and cons, that you can see on the slide, but fortunately, with our project, we have tried to combine their advantages, but our biggest difficulty is that there is no legal background for palate identification, so it is not yet legally included in the primary identification method. On the next slide, we focused on the market in Europe, so our market is mainly driven by illegal migration, with nearly 4 million illegal border crossings since 2009. And it is testing the authorities every day. So the worst part is that illegal migration can bring in people we know little or nothing about. That is why we have started to develop software that can even be used for ethnic profiling if we have enough information. Palate identification does not require a high level of background knowledge. It is easy to sample, and it is not a vulnerable area, and it is unique to each individual, including monozygotic twins as well. On the next slide, that is why we have a new solution for human identification. Can you please start the video as well? If it's possible, but if it's not, it's okay. So our software is protected by 2 patents. The point is to analyze 3D patterns of the palate to draw the palatal ridges, which are unique, and this has been proven by many research at Semmelweis University. We are looking for a collaborative partner and investor to take the development and research forward. And what you cannot see on the video, but trust me, you can see the Palatoprint software draw out the palatal ridges in blue, red, and green, and it is only on the MVP level 1. On the next slide, about the competition on the market. So currently, primary identifiers are the biggest competitors. Fingerprint has been used for a long time, but has many drawbacks. These depend heavily on dental records and the dentist as well, trust me. And DNA is a very reliable but expensive analytical solution. If we succeed in getting the palate accepted by the profession as the 4th primary identifiers, we will be very happy. On the next slide, here's the ask. Please help us and see the potential in our innovation. To create the marketable solution, we are looking for around 9 to 15 million euros over a period of about 4 years, which will cover R&D, sales, marketing, and IT development as well. We have managed to add 5 intraoral scanners to collect data, but in total, we will be able to start developing artificial intelligence on 10,000 pieces of data. We would also like to ask for further help in this. And if we have enough financial and human resources, we could have market-ready software within 12 months. On the next slide, you can see our team. The project leader on the left is Professor János Vág. Our company, our own companies, Crunch and Semmelweis University, are jointly managing the project, and the additional experts from scientific and forensic side software development strengthen our team. And I hope you have every information to make a decision. And if you have any question, feel free to contact me, and you can see on the slide next to my picture on the right. And on the next slide, here's the farewell slide. Thank you very much for your kind attention, and have a good afternoon. Bye-bye.
Thank you, Botond. Thank you very, very much for your presentation as well. Our next speaker is Ramūnas Grigonis. He is Innovation Manager at Vilnius University. Ramūnas, good afternoon. Welcome, and please, the floor is yours.
Yes, thank you. Hello, everyone. My name is Ramūnas Grigonis. I'm Innovation Manager at Vilnius University Innovation Office. And now I would like to very briefly present you Vilnius University as your business partner. Next slide, please. Firstly, a few words about our university. So Vilnius University has a very rich history since its establishment in 1579. And now Vilnius University is the largest university in Lithuania with more than 3,000 academic personnel and more than 24,000 students. And according to current QS World University rankings, Vilnius University is among top 400 world universities. And next slide, please. Vilnius University focuses on excellence in research. And a very good example of such scientific excellence in the field of life sciences is Professor Virginijus Šikšnys and his research team, who are pioneers of the CRISPR-Cas9 genome editing technology. Professor Šikšnys was awarded with very prestigious prizes such as Warren Alpert Prize, Novozymes Prize, and in 2018, the Kavli Prize, together with two other prominent researchers, Jennifer Doudna and Emmanuel Carpentier, who later in 2020 were also awarded with the Nobel Prize for the development of CRISPR-Cas technology. Next slide. Vilnius University is open for various forms of R&D collaboration with industry partners, such as providing contract research services, licensing technologies developed at Vilnius University, collaborating on joint projects, providing open access infrastructure and services, and others. And our income from licensing and research contracts with the industry within the field of life sciences is about 2 million euros per year. Next slide. In Vilnius University, there are more than 50 research teams working in fields of biotechnology, biochemistry, medicine, and other life sciences. And these research fields include epigenetics, diagnostics, microfluidics, medical devices, microbiology, immunology and antibody development, and a lot of others. Next slide. Our open access R&D infrastructure and services include the high-speed atomic force microscopy, cryotransmission electron microscopy, animal laboratory and in vivo testing, chemical synthesis of organic compounds for industry, and many others. Next slide. And here you can find links to the information about our research teams and what they offer. Then a portfolio of Vilnius University inventions and technologies that are available for licensing. Then catalog of open access infrastructure and services. And then information about university startups. And next slide. And finally, this is the contact information. So let's contact us. We'll be looking forward to it. And thank you for your attention.
And thank you, Ramonas, for your presentation as well. Have a good afternoon and good evening. Thank you. Next 5 minutes go to Robert Zorek, who is professor at Institute of Pathophysiology, Faculty of Medicine, University of Ljubljana. Robert, good afternoon and welcome.
So thank you very much for this introduction. I would like to present our research program team that deals with precision medicines. We have 2 lines of the research, it's oncology and urology, and today I would like to address oncology, an advanced personalized cell-based medicine to treat cancer that we have developed. And here is the team. I'm the head of this National Core Research Program that is carried out by our lab at the medical faculty and by a tech company, the Tech Park in Ljubljana. So our research team actually arises from biophysics, from cell physiology, and the main aim is to understand membrane fusion. We also operate the Carl Zeiss Reference Center for confocal microscopy since a while. We published over 200 papers. We have 4 international patents, and then we decided to go for a translational development. And here we have one strong project called identifying small molecules for neurological indications, a project we call Smart Cure. And also the other one is developing of advanced cell-based medicinal products, somatic cell immunotherapy, HybridCure, that actually is already now in use for patients. So in 2000, we actually established a spin-off company, Silsa Biomedical, and it's a very small company employing PhDs mostly, operates a quality management system, has a GMP clean room facilities, and is regularly audited by the national agency. Now, HybridCure is basically a cell vaccine. It's a immunohybridoma production drug, ATMP, that actually targets prostate cancer. So we take a biopsy, and then we make tumor cells in suspension. We also take sort of blood monocytes, and we generate dendritic cells, and then we fuse them with a custom sort of developed sort of technology, electrofusion. So we fuse cancer cells and dendritic cells, and then we get immunohybridomas that are back injected in intradermally, hopefully then activating the immune system in the body of the patient. Now, the clinical results, so this technology has been developed, and the clinical results actually show very promising prolongation of time to next treatment. That's highly significant, and also a very powerful correlation between survival time of patients and a change of peripheral blood cells, NK cells that are measured. So this gives a prognostic feature. So in summary, this personalized immunotherapy is already available in Slovenia. It's produced under non-routine production and allows the tech transfer to any of the countries. Our SmartCure project, however, is basically at the proof of concept stage, and it's based on my molecules that actually target aerobic glycolysis in neuroglia. And basically, my last slide is actually showing some of the technologies, how we produce these immunohybridomas, where we show cells that are fused to nuclei here, and of course, fused lysosomes. Not only 2 cells, dendritic and tumor, but also their lysosomes are needed to actually perform the antigen presentation and the immune modulation upon injection of this construct into the patient. For the contact, please contact us if you're interested. We'll be very happy to actually address any questions you have. Thank you very much.
Thank you very much, Robert, for your presentation. Have a good evening. Thank you. We have 2 speakers left, 2 presentations, and first up will be Margus Lepik, who is Vice Director of Institute of Molecular and Cell Biology, University of Tartu. Margus, good afternoon and welcome, please.
Good afternoon. I am Margus Lepik from University of Tartu, Institute of Molecular and Cell Biology. Next slide, please. So, yes, our institute has very wide range of competence. We have HCS, that are involved in teaching and science. We have great symbiosis of bioinformatics, biotechnology, cell biology, developmental biology, evolutionary biology, microbial and biochemistry, genetics that involves also microbiology and molecular biology. We have coordinating 5 study programs: gene technology in bachelor level, molecular bioscience and biomedicine in master level. Biomedicine we share with Faculty of Medicine, molecular and cell biology and gene technology in doctoral level, and gene technology we share with Institute of Genomics. And more about us and our infrastructure and competence can be found from our website. Everyone is welcome to visit the website. Next slide, please. What we are offering, basically future employees that who have PhD degrees. We have very well established the enterprise practice subject. Many of our students go to different companies. We have more than 20 enterprise practice partners. Some of them are mapped in the map here. And most of them are in Estonia and also abroad, including Thermo Fisher, L'Oreal, Solis Biodyn, Ecosagand, and so on. And if companies find very attractive and capable student, the students can make their master thesis in a company. All our master theses are based on experimental work. And if company has some project that can be defended in our institute, we can share the master students and they can defend in our institute. And we this year are going even further. We are launching at least 2 new joint doctoral students with biotechnology company. And if they are defending, then the company has new employees who have PhD degrees, but they have 4 to 6 years already worked in a company, which is very, very big advantage for the company. So next slide, please. So what the collaboration gives is access to our institute's competence and infrastructure, which doesn't mean that all the collaboration has to involve students. No, they don't. If a company has some kind of project and we have the know-how, please contact us. We will find the most suitable scientific competence from our institute and we can collaborate. We take seriously confidentiality. We signed a confidentiality agreement. The company presenter are included in our defenses and all the paperwork students sent to us are previewed by the supervisors in the company. And also we collect the feedback from the companies to develop our study programs to have the best fit for the industry. Next slide, please. And how it all happens, if company has some practice positions or collaboration of in mind, then they can contact us. We will announce to the students or our workers their projects. The companies are allowed to see the candidates' academic performance and they can talk to them and they can take to the practice all the suitable students. The tripartite agreement will be signed and practice in the company. Next slide, please. And with this, I will finish. We will forward to your contact to us. Thank you.
Great, thank you. Margus, thank you very much for your presentation as well. I hope all of you who are with us are feeling very, very good. And we have the last presentation of this session. And the last speaker is Stefan Busnatu, who is medical doctor. He is cardiologist at Băgădăsar Arseni Emergency Clinical Hospital. Stefan, good afternoon. I hope you have joined here and you can see and hear me as well.
Hello, Martins. Yes, I can hear you. Thank you very much. So it's the last presentation and probably is the newest center around all the presentations that have been held. As Martins mentioned, we are from Bucharest, Romania, and we are representing the University of Medicine and Pharmacy Carol Davila from Bucharest, speaking today about the Center for Innovation in eHealth that has been founded since 2 years ago. Our Center of Innovation basically is a structure that is aimed in developing the skills and competences for students and doctors in the digital health activities. We aim to facilitate participation in collaborative private and R&D projects to increase and to adapt to the digitization of education and health system. Basically, we are aiming to increase the collaborations that we have within the university for all medical and pharmaceutical specialties, to develop digital and innovative solutions for the medical sector that could serve all the stakeholders. Why? Because within the university, we have 37 clinical hospitals throughout Bucharest. We are having somehow between 6,000 or 10,000 medical resident doctors in preparation, over 1,000 doctoral students at the PhD doctoral school, and over 12,000 students. So we have a lot of working forces, to say, in order to involve them in different activities. Basically, the center aims to aggregate all this capacity into some core facilities that are nominated in the following way. Basically, we have a core of digital solution and innovative technologies that it's looking forward in increasing the e-health education, to propose models for digital solutions in assisting and monitoring patients with medical conditions, to test and pilot these solutions for patients, and to participate in European projects with these solutions. Another core is the one related to big data analysis. We aim to test big data solutions, basically to analyze health data and costs related to medical services, and afterwards aiming to use these solutions in the core of artificial intelligence in health in order to develop smart solutions based on the needs of the medical staff in order to improve the patient management and also to improve the medical services. Other core activities are related to space medicine, medical simulation, and innovative medical learning, and the part related to entrepreneurial hub. What we need at the moment and what we could support, the idea is that we aim for private partnerships. We already have partnerships with big pharma companies like Pfizer, Janssen, AstraZeneca, which support us basically with their technologies in bringing new educational models and e-health training courses for our students. We also are looking forward to have more partnerships in terms of technological partners for developing and testing of digital solutions, because as we told you, we have 37 clinical hospitals that could be used as testbed clinics. And basically, somehow, we aim to support the startup ecosystem development because we are enrolled now in organizing the biggest hackathon in e-health from Romania, together with Janssen. At the governmental level, we can say that by being the biggest university from Romania, the medical university, we have a position from which we can support regional and national e-health ecosystem development together with our partners. This is a series of projects in which we are involved, like Horizon 2020 and FP7 projects that are related to telemonitoring, that are related to data transferability within the European context. Just an example in order to have a glance of what we are doing. And this is the contact point in case you want to contact us. Thank you very much.
Stefan, thank you very much for your presentation and have a good evening as well to you. 12 speakers, amazing. This is it for this session for pitches of Alliance4Life's members to industry and ESRs. Thank you very much to all the speakers today. However, before we end, I would like to welcome once again Alliance4Life coordinator Zlata Novatnik, who will give a nice wrap-up of this days. Zlata, welcome again.
Thank you very much, Martin. I was asked to make a wrap-up with several closing words. So, dear participants, colleagues, speakers, we closed the first day program with the hope that it was a good and interesting day and that it opened your curiosity to learn even more. Many thanks to speakers of the first session Life After PhD, which was perfectly prepared and moderated by Esther Jarur. We heard 4 inspiring personalities who started in academia and then entered the world of business. This session was a lot about motivation, and we received a very nice feedback from participants. Like, everything said today was so useful, or it was outstanding, very inspiring. I have more confidence now. So we can also mention several take-home messages that our early stage researchers received, like change is only possible outside of comfort zone. Don't be afraid of taking decisions, or surround yourself with people better than you and learn from them. And I would say that these messages are useful not only for young researchers, but to all of us, including the management of our research organizations and of partner companies. After lunch, we heard pitches of 7 companies that are looking for collaborations and for smart colleagues. And I especially liked remarks about the needs of the industry when collaborating with academia, about skills that are needed for the industry, like project management, budgeting, writing. And it's very useful for us as we plan to organize Skills Academy and offer trainings in soft skills. So we will have these recommendations in mind. And the second session this afternoon provided information about the offer of the Alliance members. So I hope both pitching sessions opened opportunities to match the demand and the offer, which is the most challenging part. And then the communication will continue also after this event. So thanks a lot to all participants for being active, asking questions and so on. We want to make the day as interactive as possible, which is a challenge in the online space. But we recorded the sessions and we will make them available on the website so that you can check it also later. And let me also appreciate contributions of our today's moderator, Esther and Martin. Well done indeed. Thanks a lot.
Thank you very much.
Yeah, you're welcome. And tomorrow our event continues for early stage researchers. So join us again for 2 morning sessions. The first one will be about communicating science and starts at 9 o'clock. And then the second one will be on funding. So that's all for today. And may I suggest we can turn on our cameras or mics and just wave or say goodbye. Have a nice evening and goodbye. See you tomorrow. Bye.
Thank you, Zlata. Thank you for this wrap-up. This was a nice wrap-up for the first day. Thank you very much. Thank you for this amazing day from me as well. And yes, let's meet tomorrow. Have a good evening. And remember, never stop learning. And I like we can wave and say goodbye. Thank you very much. Have a good evening. Bye.